Question 7. Scientists studying the nucleus of the fruit fly, Drosophila melanogaster, observed distinct types of nucleic acid chains. These scientists noticed that one type of nucleic acid chain was able to pass through the nuclear membrane and move to a ribosome. After the nucleic acid chain attaches to the ribosome, a polymer is produced, and we know that polymer should be a polypeptide. Describe the steps occurring at the ribosome that resulted in the production of the polymer. What are we after here? We're after translation, because we know translation occurs at the ribosome where protein synthesis occurs. And so basically what we want to give them are the steps to translation. Now, students are normally really good at describing translation, but I think the big thing is just to make it really specific and concise. You clearly see that that is the real agenda of this walkthrough. It's to give you clear, concise answers, because I guarantee you students would have probably filled up this entire thing. Obviously, that's dependent upon how big you write, but again, dot points will really help you get everything across to them. So we know there's 3 steps to translation. We know there's initiation through the start codon, then we know there's elongation, and then there's termination. And the big thing here with translation is we want to go through these steps. So first and foremost, we know that the mRNA, the messenger RNA, binds to the ribosomes, and the ribosomes use this mRNA code. They read it, obviously 1 codon or 3 nucleotide bases at a time. Then we know that tRNA brings a specific amino acid to the ribosome. And then the tRNA anticodon joins to the complementary mRNA codon. An amino acid is added to the polypeptide chain through peptide bonds, not necessary in the explanations from the examiner's report, but there for completion. And therefore there's termination. A stop codon is reached, indicating completion of the polypeptide involved. Again, this is, in terms of rote learning, a very, very similar premise when we're looking at explaining and articulating ourselves. If you can go through this in the same way I described PCR in a later video, then really you'll absolutely zone in on what you need to know for translation. You can do the same thing for transcription, but these are the above steps that I would go through. Then we move down into B. And then it says 1 particular length of nucleic acid passed through the nuclear membrane and coded for the production of a polymer. Well, again, we know that polymer is the polypeptide chain that was 90 monomers long. How many nucleotide bases on the nucleic acid chain were involved in the coding for this polymer? And explain our response. So remember, we know that there's 3 nucleotide bases that code for 1 amino acid. And then we'd go through this, and the understanding is there's 3 to 1. We know if there's 90 monomers, there must be 270 nucleotide bases. And so that's all you really need to do with this particular question, is say there's 3 nucleotide bases that code for 1 amino acid, and therefore a polymer that is 90 monomers long would have 90 times 3, which is 270 nucleotide bases, which is your final answer. I just want to go back to this because it's just struck me now that with A, you could have also, with B, you could also say 273 and 276. The reason why, if you did 273, there would obviously be a start codon, and 276 is when there was a start and the stop codon. So 270, 273, and 276 were all accepted, and students actually did this quite well, with 40% of them getting full marks.